feel pretty comfortable where we are right now.
Okay. I saw that you did the assignment.
Yes, I was able to get that completed.
Super. And you still have the discussions from last week you need to get taken care of.
Okay.
So what we'll do today is do assignments or do questions and just review a couple of things and then do the in-class activity.
Okay.
And then we will, from there, we will take some time to let you guys continue to work on the assignments, assessments, etc.,
Okay.
and the discussions. So, yeah. So we've got all of this going on today. So we're getting to be halfway through the class. And I'll probably do a little introduction to next week's major topics because we're going to start switching over. We're going to do some more dosage calculations of a more, I won't say complex, but a set of calculations that require some different considerations, which are flow rates for infusion of drugs. So we'll talk about that after before we do our in-class activity and then go off to other things. So
Okay.
that's the plan for today.
Perfect. That'll be really interesting because we touched base lightly on dosages that with the like IV drips and stuff like that, but we didn't go into much detail. So I think it'll be interesting to learn a little more about that.
Yeah, a lot of drugs are given over time. Plus, if you are one of the first things that happens when someone goes into a clinic or hospital is, and they have a problem, is they may be set up with an IV. So you need to know not just the technique, which is not what this class will teach. In fact, you haven't been taught how to set an IV as far as I know. But that's something that you can be trained on when it's necessary. But just being able to understand that we don't just set an IV and turn things on. We are given clear instructions as to how much drug is to be given per unit time. And then we have to calculate either what the settings are on an infusion pump or how to set up a what I call a manual delivery system, which basically uses gravity to put drug into the patient. And that's a slightly different calculation because we have to calculate how many drops per minute the patient is going to receive. So it's a little different. The infusion pumps work in milliliters per hour.
Oh, wow. Okay.
So we'll get some basics on that today, later on. And I'm going to just quickly ping Michaela. Okay, that's done. Um, so let's just do a quick review here so that we can remember a few things for the assessment and for the in-class activity.
Okay.
Um, First off, I think I said this before. Ideally, every drug given to every patient would be tailored to the specific patient. However, that's just not a practical consideration. Um, so, drugs that are given orally, generally speaking, are developed in what we call a fixed dose approach, which is if you are dealing with an adult. And we, as I said before, our clinical trials use define adults as about 18 to 55 or so. And the reason is um, that we start at 18 is that's the age at which people can sign the consent forms to actually participate in a clinical trial. Um, Anyone younger, a parent or guardian has to sign those forms. And there are some there's some sticky issues of being careful about how we get parents or guardians to do that. I said we had developed a drug for premature babies, um, and that was to help their lungs inflate because the lungs and the digestive tract are the last systems to actually develop in a newborn for the simple reason that in utero, they don't need them.
Right.
So, but they come out hearing perfectly well because they've been hearing their mother and the people around the mother all during the gestation. But um, those two systems really are the last to develop, and they usually develop in the last few weeks of pregnancy. So when a baby is born significantly early, that baby is needs to be supported. This one of the things that is a real problem with extreme premature birth is that they pass away. They die because they really can't breathe. It's hard to get their lungs to start functioning properly. So it can be difficult. The environment in a neonatal intensive care unit is really stressful. I admire the nurses and doctors who can work there.
I do as
Um,
well.
but what happens is we develop this drug for helping inflate the lungs of these newborns, these premature babies. And but of course, in order to enroll a newborn into a clinical trial, the parents have to agree. They have to sign that consent form. Well, you go to a parent and say, well, we might have a drug we have a drug that might save your baby's life. What are most parents going to say? Yeah, give me the
Yeah.
papers. That's what they're
Yeah.
going to say.
Oh yeah.
So you have to be very careful in approaching them that they understand that you're not guaranteeing results and trying to get that through that filter of, oh my God, my baby might die if I don't do this.
Mhm.
It's a difficult process. Um, and so we had quite a time with that.
Oh, I'm
I,
sure.
I have a slightly funny story to tell about that drug. I was the project manager for the development of that drug, so I worked a lot with the clinicians and and everyone else involved in it. So I knew the drug pretty well. And at the time we had it in clinical trials, our second child was born 7 weeks early. So she immediately went into um, the NICU. We were living at the time in Durham, North Carolina, so she was taken into Duke, which was one of our clinical trial sites. Um, so it's it's midnight. It's actually 1 o'clock in the morning, a little after 1 o'clock in the morning on New Year's Day.
Oh, wow.
And so we'd had trouble getting the Duke folks to answer the phone from our county hospital that we were in at the time, when we realized that this pregnancy was, or this birth was not going to be postponed. This baby wanted out. Um,
Oh,
and so we had spent a lot of time trying to call over to the Duke system, and they wouldn't answer the phone. Now it's New Year's Eve. We had our OB was in the county hospital with 10 women in childbirth, in labor.
oh my goodness.
Yeah, well,
Oh my goodness.
stressful time. So we got the
Yes.
baby, we got her transported over. She was actually born in the ambulance between the county hospital and Duke. But we got there, and there she was in the facility, and I walked in, and it's New Year's Eve. We hadn't been partying because at that time we weren't doing that sort of thing because of my wife's pregnancy. So we hadn't been partying, but there I am. I'm wearing a flannel shirt, probably mismatched socks, looking kind of disheveled. And so we've got these little wannabe doctors pulling clinic duty while they're in medical school, and
Oh,
they're sort of casually fussing over our baby, blowing oxygen into her face. And I look at him and I said, well, is she eligible for the Exosurf protocol, which is the Exosurf was the drug. And all of a sudden, this party atmosphere just went away. They got deadly serious. All of a sudden, they didn't know what to think of this guy who knew about an experimental drug. And the next day, so they said, well, that sort of response, we'll have to find out. So the next day, I came in and was visiting with Charisse, our daughter, in the NICU, and in walks the director of the NICU, and oh, she was really serious. How do you know about this? And so we talked about it for a while. I told her that I was the project manager and I was working with the lead clinical investigator at our company. And so then she went through this incredibly lengthy description of why she wasn't eligible to be treated with the drug. She didn't need it, it turned out. She was
oh,
more developed, even though she was that early, more developed than any other child in the NICU. She was the healthiest baby there.
well, that's some relief.
Yeah.
I could imagine. Oh my goodness.
But the expression on this doctor's face was really interesting.
How
And
do
I think
you
she
know
got better
about this?
care. I think she got better care because of it.
Well,
So,
you
which
would
was too
hope they
bad.
all receive good care, but I mean,
Yeah.
when you're showing that you know about something, and I mean that's not even really I'm sure it wasn't discussed much outside of, because were you at trials yet with
Oh yeah.
oh, okay.
And the Duke NICU was one of the trial sites. They were helping us with doing the clinical trials on these babies.
Okay.
So they knew
Okay.
this
Well.
drug. And I think the lead Duke investigator was the woman who came in and talked to me the next morning. How do you know? Okay, well, here's how I know. So anyway, we're fine. She's fine now. She's our daughter living in Denmark, and is about to have her third novel published. So she's doing well.
Oh wow, congratulations. That's
So
great.
yeah, so that's those are some of the things that have to be considered though when you're developing drugs in young people, in patients. Now, it's at the other end of the spectrum, you get to your geriatric patients. Very clearly, the elderly can sign their consent forms. But one of the things you have to be careful with the elderly is that they're mentally competent to do so. Because of
Correct.
course, a lot of elderly have cognition problems. You have to be sure they understand everything fully and are considered competent to sign a consent form. And if you don't think they are, you can't enroll
Okay.
them. And
That makes sense.
yeah, part of the consideration there, of course, is that if something goes wrong, if they don't respond to the drug, or if they, God forbid, have an adverse reaction to it, then you've got issues with family members coming in and wanting to sue.
I'm sure.
So, so you have to be careful with with many of the populations we deal with. Drugs in older children are a little easier because unless it's a life-threatening situation, again, it's not. You still have the same considerations. The parents need to sign. They have to understand what the risks are, and what the potential benefits could be. I mean, obviously, all drugs are a risk-benefit consideration, but you have to be sure that they understand these things. Children, as I say, are not quite the same level as a premature baby who might not survive the day.
Right.
But, there's a lot of kids that have cancers.
Oh, yeah.
And we have remarkable success in treating those pediatric cancers, the cancers in children. But that's a totally different set of stories. But we have to be careful that when we're talking to parents, that we don't make promises that that are not possible to know we can keep.
Right.
So it's a delicate thing. With adults, I mean, you can explain it and make sure that they sign it, but you also need to be sure that they understand it.
Mm-hmm.
And as a result, there's a lot of scrutiny put into consent forms and to how clinical trials are conducted with drugs. But what is the purpose of a clinical trial? Well, when you saw the video probably of Elixir of Death.
Yes.
Yes.
That is the perfect example of
Yeah.
why there are clinical trials.
Exactly.
My grandparents, my grandma remembers that.
Oh, really?
Yes. She remembers that with her. She said her and her brother never had to take it, but they remember hearing about it.
Mm-hmm.
And I was amazed with that movie that doctor was able to do that.
Yeah. And
But
that's really, the FDA existed. The Food and Drug Administration actually existed, but it was kind of a weak agency, had very, very little real enforcement or control powers. And that 1935 incident led to the structure that we have today for developing drugs, because the Congress, under pressure from a lot of people, passed a law that said that the FDA had to approve drugs based on clinical evaluation for safety and efficacy. So they had to be safe. We take that as a given now, but that wasn't always the case. In fact, the reason there are any drugs at all is because of late in the 19th century, a lot of stuff being sold that were actually very, very dangerous. One of my favorites is Lydia Pinkham's elixir. And Lydia Pinkham's elixir was something what was sold to women to deal with what they referred to as ladies' issues at the time, mainly things like PMS and that sort of thing, what we
Okay.
now call PMS. But it was a mix of alcohol and opium.
Oh, lovely.
Yeah. So yeah, you take a hit off that bottle and you go from feeling sad to feeling very happy.
Happy.
Um, but they're dangerous drugs, and so opium was being targeted. Opium was a major problem in this country at the beginning of the 20th century,
Oh yeah.
and a big problem in the UK and Europe because the opium trade was a major part of building the British Empire. Um,
Oh,
and in
okay.
fact, the opium trade led to 2 wars with China, between Britain and China, the Opium Wars, because the Emperor of China wanted to ban it. It was a problem for the Chinese people. He wanted to ban it. And the British said, no, you're not, because that's going to cost us money. So they're another
Oh,
good example of money more important than people.
yep.
And so 2 wars were fought with China to keep the opium trade going. Opium, that was in the middle of the 19th century. Got to the point where opium was a major problem. In fact, when Arthur Conan Doyle started writing stories about his fictional detective, Sherlock Holmes, when Holmes wasn't on a case, he was smoking dope,
Oh,
smoking opium. It
wow.
was a major issue at the time. So, and during the latter part of the Victorian era, the reign of Queen Victoria, they banned opium dens and stuff from the UK, and it came into the United States, and we started trying to ban the use of opium. So Lydia Pinkham's was basically taken off the market after the first Food and Drug Act in 1906. But
That's good.
then, yeah, but then the FDA wasn't given that strength to really regulate the development and marketing of drugs until 1935. The emphasis has always been on safety. When we develop drugs, the first set of clinical trials are what we call safety trials, and we use what we call normal volunteers. And you might argue that anybody that lets an unknown substance be put into them without having any idea what's going to happen isn't normal. But what we mean by that is that they don't have a disease. They don't have the disease that's being treated. So we wanted that strictly to evaluate how safe the drug is. And a lot of
Okay.
drugs won't survive that experience because we can only predict based on animal studies the potential for toxicity of a drug.
Okay.
But we can't, until we actually put it into people in a very carefully controlled manner, we cannot determine whether or not that's true, because even though there are certain people that we probably would argue about, we're not rats.
Right.
So, so we don't respond the same way.
Right.
And there are rules against experimenting in primate species, monkeys, rules and extreme costs that make it not, obviously we would, if we really wanted to know if a drug was safe in a human being, we would put it into a chimpanzee, our closest genetic relative in the animal kingdom. But we can't do that. It's, first off, they're sort of endangered, but the second off, they're very, very much controlled in terms of access. It can only be done under extremely rare circumstances. However, that first trial determines what dose we can give a patient before they start showing toxicity. Most of the times that toxicity is completely reversible. It'll be some sort of liver enzyme elevation or a change in creatinine clearance time, which indicates kidney issues. So so we look at those, we look for those things. And at that point, we quit giving the drug to the patient. We don't take the dose any higher. And what
Okay.
we've done in our animal studies is based on studies in animals, we do our best to determine what a likely therapeutic level in plasma of the drug would be. And the whole idea is that when we determine the toxicity of this drug, when we start seeing a toxicity, it ought to be orders of magnitude above that therapeutic dose.
Okay.
That's our goal. Is it always the case? No. Do we have drugs out there that are have what we call a narrow therapeutic window? In other words, difference between the effective dose and the toxic dose. Yeah, one of the most prescribed drugs we have, digoxin, falls into that category. It has a narrow therapeutic window, but it's one of the most useful drugs we have for treating various cardiac conditions. So we use it very carefully. And I had a young cardiologist neighbor when I lived in North Carolina who told me that it was his absolute favorite drug because he could use it. It was well understood, had been studied, had been being used for between 200 and 300 years. And it was well understood. And they
Okay.
knew how to dose and they knew how to deal with it. And when somebody got an overdose, we actually had a rescue drug.
Oh,
So,
wow.
so it was really, it's just one. And every time somebody's tried to improve on the drug by developing a synthetic drug that's supposed to replace it, it hadn't worked. So
Wow.
that's been around. It's actually an extract of the poisonous plant foxglove, extract of the leaves of foxglove. So, because we cannot, it's so complex chemically, we can't manufacture it.
Wow.
So, and it's cheap, very
So
cheap.
do we farm that foxglove now then?
Yeah, there are people
Okay.
who actually grow foxglove, harvest the leaves. They grow it using very specific techniques so that we don't, get things into the leaves that we don't want.
Okay.
And then there's a very careful extraction process that people use. That was another drug that my company that I worked for made, and our extraction process that we used, we never patented. We maintained it as a trade secret because it was unique. I have no idea what part of it was so unique, but we had a very unique way of extracting it that gave the highest quality version of digoxin that's on the market.
Wow. That's interesting.
Yeah. Yeah, so that's been a trade secret for well over a century. Patents last 20 years. Trade secrets last as long as you can keep them.
Wow.
So,
That's amazing.
but I saw the raw material. We had one of the places we did the extraction was at our manufacturing plant in North Carolina, and I used to go there regularly for meetings, and I'd see bales of these leaves come in
Wow.
on a truck, and they'd put it into this black box room that only a few people were allowed to go into, the people
Oh,
who
I'm sure.
understood the process, and the rest of us were kept out.
Not a place you'd really want to go, though,
No,
especially if it's poisonous.
yeah. No, you have to be real careful. The guys going in were well protected.
Oh, I'm sure.
Not just your average hospital PPE. They
Right.
wore very high level protection because of the toxicity of the drug they were working with. But anyway, we have gone a little astray here, but that's okay. The idea, then after we determine that the drug is safe enough to give to humans, and as I say, a lot of drugs don't survive that experience. In fact, probably, you know, we will, you know, for every drug that goes on the market, we probably investigate between 15 and 20,000 potential drug candidates, because these things, you know, drugs are toxic. You know,
Oh
they're
yeah.
poisons. The body doesn't like them. So in some cases, those poisons are real, and you know, and most of the candidates. I mean, when I spent my time, I started off as a chemist in the pharmaceutical industry, making drugs, potential drugs. And I probably, this was back in the days before chemistry changed and you can make thousands at a time. At that time, we made one at a time. So I'd probably make 120 or so a year of purified substances, new substances to be tested. And exactly one of them got into clinical trials and it didn't survive.
Oh wow.
So, and that's typical. I mean, and in fact, there are many people working in the industry at that time who never had anything go that far.
Wow.
So it's a risky business. That's not a good excuse for the high prices of drugs, but it is a risky business. But we, so the second, after we do that, that's the second set of clinical trials is when we determine dose, effective dose in patients, and the schedule that the patients need to receive the drug on.
Okay.
Meaning how many times a day and what the dose, you know, the level of the dose. And this is all done in, again, a gradually escalating sequence of doses to actually measure the efficacy. Again, we have an idea of where it should be from studies in animal models, but again, people are not animals. We
Right.
are a little bit different. So um, we have to do this very carefully. So this second trial is what we call a double-blind clinical trial. That means that neither the patients nor the clinical investigators on site know whether they're getting active drug or placebo.
Oh, okay.
Okay, and in fact, we've actually moved. It's actually moved up the chain. Over the years, we've moved into something that we might call triple blind, because we use randomized. We use a computer randomization to determine whether a specific numbered dose has active ingredient or placebo. And even our clinical staff on the inside of the company can't see that. So
Okay.
we don't know what they're getting. The physician on site doesn't know what the patients are getting, and the patients don't know. And there's part of the consent. The patients are told, you might not get active drug. You know, there's a good chance of it. In fact, ideally you'd go 50-50, but depending on the drug, we've done different things. For really dangerous drugs, we developed the first treatment for AIDS, in fact, and there what we did was the drug itself was so toxic that our clinical trials were comparing low dose versus high dose drug and determining based on that. And again, but we didn't know who got what. So what happens is now we've established, the companies will establish what we call a clinical review board or an institutional review board of people who are not company employees, people who are not involved in the clinical trials. These are biostatisticians and physicians and other experts in the field. And what they do is monitor the results. They too don't actually know who gets what, but they know that there is a group, call it a group A, a group B, and a group C of patients. And what they're doing is looking to see if there's a differential in either response or in toxicity between the groups. They don't know who gets what. So when they see what their statisticians tell them are a statistically significant difference appearing, they will stop the trial. They have the authority to stop the trial. And at that point, we break the code. And of course, we hope that what we're seeing is that we have a group that's getting better and they're the ones getting the active drug.
Okay.
But that's what we do with that. So we try to keep it as objective as we can. Is it perfect? No. There's a lot of discussion as to our dependence on this, what we call the well-controlled clinical trial, but we don't really have a better way of doing this. So, but that's how we do that. And once we have determined that dose, then we move on to giving large populations, enrolling large numbers of people in studies to look for, basically to look for more information on safety and efficacy in population of ill people. In that second trial, we generally want people with only the disease we're treating.
Okay.
The third trial is supposed to simulate true clinical practice, where somebody walks in, they're sick, they've got 17 other diseases, they're going to get the drug. So is that a good idea? We don't know. And what are the interactions between whatever drugs they're taking and your new drug? Mm-hmm. So this third trial is to really nail down long-term indicators of efficacy and, of course, long-term use safety. And again, and another consideration is there are really drugs that you take for a short term. They're acute use drugs. Antibiotics are some of the best examples. And then there are chronic drugs, antidepressants, drugs for blood pressure control, et cetera. So we have the clinical development of an acute use drug is shorter than for a chronic drug, because we have to, in the chronic case, we do have to have data out for 2 to 3 years of patient use. Because that's what clinically they're going to experience, or what they will experience when the drugs are available for widespread use. So it's a complex issue. And then companies keep the studies going even after the drug is approved, because they really have to monitor safety issues.
Oh, yeah.
Good examples would be the effort to try and replace aspirin with aspirin affects an enzyme known as COX-1, cyclooxygenase 1, in its ability to reduce inflammation and cause pain and fever. So there is another variant of that enzyme, cyclooxygenase 2, and Celebrex and several other drugs went on the market that were supposed to be much safer versions of aspirin. And they looked good in their clinical trials until they were on the market, and then we started seeing people die.
Yeah, I remember this.
Right. So Celebrex stayed on the market until fairly recently, because there was a certain class of patients that really needed a COX-2 antagonist to respond to certain conditions, but it now is gone.
Oh yeah, you can't find it on the shelf.
Yeah. So so there's lots of stuff that goes on in this development, but we determined in that second set of clinical trials the size, the potency of each dose, and the frequency that we have to give to the patients in order to achieve what we hope is the best result for our patients.
Okay.
So that's what we are. That's what we do in the clinic. Now, where does that come in? Well, that clinical experience tells us a couple of things. When we're looking at adults, it allows us to set a dose for a dose that is going to be effective for the vast majority of adults. And of course, adults range all over the map. We've got, we've got the jockey, the 98-pound guy or woman. There are women jockeys now too. But we have the 98-pounder adult who's, 25, 30 years old. And then we have people ranging up to many hundreds of pounds. And so what we want is a drug that's going to be effective in most of them. The really, really large patients may need more drug, but it all depends. Even the very, very large patients, most of us have about the same plasma volume.
Okay.
About 5 liters. It doesn't matter if we are the 98-pound jockey or some person that weighs, 400 or 500 pounds. Basically, we have that same volume that we're dealing with. So though that's one of the reasons that works. But children, and especially babies, obviously don't have 5 liters of blood.
Right.
So giving them an adult dose is going to then potentially overdose them. Even drugs that have a wide therapeutic window, that difference between between effective and toxicity. We can find children being overdosed. And if we go back to, the Hippocratic Oath, do no harm. That's only a very small portion of the oath, but do no harm. Then we want to make sure that we are not, that we're not even approaching harming, especially children. So, so we have to find a way to reduce those doses. And as we discussed on Monday, we have weight or age or body surface area considerations as for a way to measure the relative size of 2 individuals. It turns out we weight and age, at least in people over maybe 1.5 or 2 years old, weight and age work pretty well up until about age 12. At age 12, we need to be looking more at the issue of something more accurate, which would be body surface area. But remember, Clark's rule was weight. That's a good measure of how large a person is. And then Young's rule was based on age. That was the one where you divide the age in years by age plus 12.
Well, yep.
And so that's why when you get above 12, it no longer is a valid calculation.
Okay.
Pardon?
Oh, I said, okay, I'm sorry, I yawned.
No, that's all right. So that's one. Okay, I haven't heard anything from Kayla. All right. So when we get to that level, when we're looking at older, then we need to look at either weight or body surface area. And then Freed's rule is for pediatrics. There are lots of little sub-rules around Freed's rule, but in general, we're looking at children about a year or younger, although it can go up to 15, 16 months, depending on the size of the child. But Freed's rule is for truly pediatric patients, babies.
Okay.
And so we want to look at that. But in reality, we know that that the best measure of size is body surface area. It's a more it's a little more difficult determination. You either have to use that formula or you have to use a nomogram
Okay.
to determine what that is. And of course, the nomogram is based on that formula. So they will give you the same results, essentially, depending on how straight your ruler is, of course. But and then, again, then the judgment area comes in when you're looking again at pediatric patients. Is someone of normal height to weight ratios? And if not, then you have one set of or one set of readings. And if so, then you have another. So we want to be sure that we pick the method that's going to be accurate. We saw, looking at both Clark's and Young's, that at least sometimes we're going to get exactly the same dose. That isn't always the case, but the difference is not going to be tremendous. So, so we need to be careful about how we choose and how we put that together. So we want to remember that we have the ability to do the calculation or to do or use a nomogram for body surface area, but the other calculations are straightforward. Clark's rule is weight in pounds divided by 150. So if we happen to be working in a clinic that weighs patients in kilograms, we'll probably have to change kilograms to pounds, which is no big deal. And in fact, in other places, I would imagine that Clark's rule is going to be based on kilograms. So we would just be dividing 150 by 2.2. So then the denominator then becomes about 68 kilos. So there's two ways of approaching that. Age is pretty clear. All measurement systems measure age in months and years. So those two are not going to change depending on where you are working in the world. So we'll be able to use those where appropriate. But again, if I had my druthers, everything would be based on body surface area. But it's just not that convenient to do that. Make sense?
Yes, that makes sense.
Okay. So it's now a little bit before 10. Let's take our first break.
Okay.
We'll come back and take a look at the in-class activity and get you started on that.
Okay. Sounds good.
So I'm going to pause the recording and mute my microphone.
Sounds good.
All right. See you in about 10.
Okay.
Resume recording here. So the week 2 in-class activity, the first line, it's about dosage calculation. The first line says, interpret the following prescriptions. Well, some of them don't need to be interpreted. They're just a calculation. But of those that you can interpret, go ahead and do so. Write it out. And that's basically to give you practice with interpreting prescription terminology. Okay?
Okay.
Okay, so what I will do is go ahead and pause the recording while you're working. If you have a question, just speak up. I will mute my microphone. Just speak up, and I will, and I'll come back on and record whatever it is we discuss.
Okay, sounds great, Mike.
Okay.
Are calculations on these, or is that not necessary?
Um, I would like you to, although typing it on might be a little iffy. But if you could, I'd love to see how you do it. But the answers are what's important for these.
Okay, perfect.
Okay.
I'll do what I can.
Thanks.
Thank you.
You're welcome.
I just have a quick question on number one. I feel like I'm overthinking it, but I also am confused by my answer that I calculated.
Okay.
So I just,
Yeah, here we are.
I
Number
used, yeah, I used the dose ordered by over the dose on hand times the quantity.
right.
And I mean, I know in the prescription it says 4 times a day, like what's being prescribed, but it would still be one tablet, correct?
Well, yeah, the formula calculates the actual dose per time you take the drug. Okay. But take a look at this. We've got 125 micrograms is the order and our 0.25 milligram tablet. So you need to convert the milligrams to micrograms, remember?
I did do that. Um, I
So
got
what,
two. I got 250 micrograms.
right.
Okay. Um, and I divided 125 micrograms from that, and I have 0.5. But
That's
that
the
would.
right answer.
Oh, okay.
Half a tablet.
Okay. I was getting confused because it was half a tablet. So okay. Thank you for explaining that.
So are you done with the in-class activity then?
I have 3 more to go that I was kind of working on.
Okay.
So that was my fourth one that I kind of was second guessing myself. So,
Don't do
uh,
that.
yeah, I just have question 3, 5, and 6 left. So,
Okay. So go ahead and work on that. I would like to talk a little about stuff related to next week before we end today. So go ahead and finish that and then we'll do that, okay?
okay, sounds good.
All righty.
Thank you.
You're welcome.
I wanted at first to use Clark's rule just because of the child's weight, but that's not the adult dosage. That's the pediatric. So I'm questioning, do I have to convert the weight into kilograms from
Yes,
pounds?
yes, you do.
Okay.
And one thing to think about, if we are dosing by weight, we don't use any pediatric reductions because it automatically adjusts for the mass of the child.
Okay.
Okay, so the purpose when we're looking at the pediatric reductions, that's when we are taking a fixed dose and looking at that. So anytime we're using something that is based on size, which would be weight, but not age.
Okay.
Not age. But weight or body surface area, then we're automatically reducing for the child itself. They can be, so this dose can be applied to any child. Notice I said it's the pediatric dose. It's actually less than the adult dose.
Okay.
So it's already been adjusted for.
Okay. So when I did calculate, as in converting from pounds to kilograms,
Mm-hmm.
I got 15 kilograms.
Right.
So with the pediatric dose being 15 milligrams per kilogram, would you take 15? I'm sorry, and
No problem.
multiply that by 15 as well
Yes,
to get your dosage for that child?
that's correct.
Okay, because I did do that, but like I said, I feel like I'm overthinking things.
Right. And really it's something that does need to be emphasized in that dosing by weight obviates pediatric reductions. So it automatically does that for us, as does body surface area.
Okay.
So, and when we do body surface area calculations, then we can use that to determine what a child dose could be by comparing the body surface area of the child with the average body surface area of a human, which is 1.7 milligrams per meter squared. That formula, by the way, applies no matter what patient you're looking at. The body surface area divided by 1.7 applies to adults as well as children, because all we're doing there is the same here. We've got 15 milligrams per kilo, but if this had been 15 milligrams per meter squared, we'd simply figure out the body surface area of this child and divide that by 1.7. That gives us the ratio of that adult weight and just allow us to calculate from there.
Okay.
Okay?
That makes sense.
Okay, great.
Thank you for explaining that.
You're welcome.
Sometimes I get in my head too much.
Yeah, it's easy to do. It's easy to overthink these things.
Thank you.
You're welcome. So, so you've done that. Have you done the others that you were working on?
No, not 5 and 6, not yet.
Okay, great.
I know Bubba's.
Yes.
I think I have it figured out to some extent. So basically, for question number 5, I converted the pounds into kilograms,
Mm-hmm.
and then I took basically the 15 milligrams per kilogram per day.
Right.
I did 15 times 20, which gave me 300 milligrams.
Right.
And then I took that and I did the dose ordered by the dose on hand times 5 milliliters.
Mm-hmm.
And ended up with 6 milliliters. And so I have to convert that into teaspoons, correct?
Yes.
Okay. I did that, but going off of my error last week on last week's in-class activity, I got 1.2 teaspoons.
That's right.
Oh, wow. I was thinking that was wrong. I thought it had to be like a whole.
No.
No?
No.
Okay. Well, let me type that in and I will get that submitted.
Okay, then we'll talk for a few minutes and end the class. So go ahead and get that submitted and let me know when you're done.
Okay, Mike, thank you.
You're welcome.